This is Christopher Sernaque hosting episode 10 of season 3 of the Current Topics in Science podcast. This podcast will address breaking scientific news in light of the origins debate and host interviews with scientists. This podcast is available on the following platforms: iTunes, Stitcher, Google Podcasts, Spotify, and iHeartRadio. Video recordings of the podcast will be uploaded to YouTube. Enjoy the podcast. Today on Current Topics in Science, we have the honor of hosting Dr. Donald Batten. Dr. Batten received his Bachelor of Science with first-class honors in agriculture and his PhD in the science of plant physiology from the University of Sydney. Dr. Batten has published over a dozen refereed papers in science journals, has done 5 agency-funded scientific research projects. He was a research horticulturalist, a senior research horticulturist, and a private horticultural consultant. He's published numbers of postgraduate theses, books, and book chapters, and he's been involved with major reports and editing. He's had decades of experience in horticulture, and his research included environmental adaptation of tropical and subtropical fruits, such as the mango, the lychee, and the custard apple, floral biology, floral induction, breeding, environmental physiology, especially with water requirements, the effects of water deficits, plant taxonomy, and mineral nutrition. Dr. Batten is now the CEO, the managing director, the senior scientist, a speaker, and a writer, and editor for the Australian office of Creation Ministries International. Now, without further ado, good morning, Dr. Batten. How was your day, and how are you doing?
Good morning, Christopher. It's great to be with you. Well, it's in the morning here, the evening for you, being on opposite sides of the world. So it's great to be with you, Christopher.
Well, Dr. Batten, it is an absolute pleasure to have you here. And of course, over in Australia, it's May 4th. So May the 4th be with you. And I hope that you're having not just a good day, but a spectacular one. And it's a great honor to have you here with us. So now, since this is Current Topics in Science, we're going to quickly look at this week's current topic. Dr. Batten, we are about to take a look at some buried treasure. The New York Times reports one of the world's oldest science experiments comes up from the dirt. The tagline reads, "Every 20 years, under the cover of darkness, scientists dig up seeds that were stashed 142 years ago beneath a college campus." The article reads, "On Thursday morning, several hours before sunrise, Majoria Weber arrived at the rendezvous spot on the campus of Michigan State University. Dr. Weber and her colleagues are the latest custodians of the Beal Seed Viability Experiment, a multi-century attempt to figure out how long seeds can lie dormant in the soil without losing their ability to germinate. Every 20 years, the experiment's caretakers creep out to a secret location under cover of night, dig up a bottle, scatter its seeds over a tray of sterile soil, and see which ones grow. It's one of the world's longest running experiments, having already gone on for 142 years, and the botanists in East Lansing hope that it will last for at least another 80. What was exactly inside the capsule? Penn Live reports, inside the bottle, the team was looking for over 1,000 seeds. 50 seeds, each of the 21 different species, from black mustard to white clover to red root armathian. Dr. Batten, what do you think about this experiment?
Well, a lot of foresight to actually have started this experiment, knowing that you wouldn't be around to see the finish of it. So, it's good stuff. It's interesting that all but one of those species was a common weed. So the one that wasn't the weed was the white clover, Trifolium repens. And one of the things that makes something a weed is that the seeds are very persistent. So, and many of the weeds have what they call hard seededness. So hard seededness is an ability to be dormant until something happens to break it out of the dormancy. And the something can be something like fire or scarification, in other words, abrading it, or cold. So a particular period of really cold conditions can break the dormancy. So the idea is that the seeds then persist over a number of years. And the question being asked by the scientist was, how many years could these seeds persist? And so he started this experiment. And I don't think he anticipated that they'll still be going now. So the interesting thing is that some seeds have incredible ability to last. I think the record of something which is definitely dated is a 2,000-year-old Judean date palm, 2,000-year-old. And it was found in excavation of Herod the Great's palace in Masada in Israel. And so it was preserved in a cool, dry place. It wasn't even refrigerated or anything like that, not by freezing. And it was germinating in 2005. So that's 2,000 years that seed has lasted. It's amazing how our Creator has put that ability in seeds to last so long. And so interesting experiment. Interesting, Darwin actually had experiments. Charles Darwin did experiments on seeds, looking at how they survive in salt water. And of course, we're interested in that because with the flood, how did seeds survive Noah's flood? And a lot of the seeds have hard seededness, which means that they're very durable even when they're soaked in water. And Darwin did these experiments on seeds in salt water to see how long they last. And quite surprising to him that some of them last many months in salt water. So he was interested from the point of view of how do seeds get transmitted from, or how is it that different continents have the same species or very similar species of plants on them? And they must have been transported across the sea. So that meant they had to be in salt water for months. And so this is the reason for his experiment. So it's actually mentioned in my Creation Answers book that we publish in terms of the distribution of plants and animals around the world today. So very interesting experiment.
Dr. Batten, thank you so much for talking about that experiment. This actually reminded me of my own time back in school. When I was in biology class a few years ago, my friends who believed in theistic evolution, they had challenged me by saying that the dark cycle is an example of unintelligent design. They said that the dark cycle is insufficient because you need 6 3-carbon molecules for the process, and only one of those 3-carbon molecules makes glucose, while the other 5 3-carbon molecules regenerate as RUBP. So I reached out to CMI and you responded. Part of your response read, ask them to demonstrate how a more efficient system would work. On the basis of their reasoning, the citric acid cycle, look it up, would be an example of unintelligent design. Ask them to show you how even 2 enzymes could evolve in concert, let alone the suite of enzymes needed for a cycle. And a cycle does not work without the whole lot present and the membrane systems to boot. Dr. Batten, I'm wondering if you could please expound on this. What is the dark cycle, and is it evidence for or against intelligent design?
So did your friends manage to show how they could improve it?
No.
Oh, dear. We hear this argument all the time, the argument of bad design. So it's actually a smokescreen for the fact that they have no idea how evolution could produce something. And Richard Dawkins used this with the vertebrate eye, claiming that it's wide back to front. And what sort of intelligent designer would create such a ham-fisted design of an eye? But of course, he and nobody else can actually explain how the reverse arrangement of the eye could even evolve. In fact, it's a huge problem with the evolutionary story. So they go on the attack and try to claim that, well, look at this design of the eye, it's so bad. Clearly, there's no intelligent designer. So they still stick to their atheism, but they argue that an intelligent designer, like the Bible talks about, would design perfect things. But of course, you can only design something which is possible. And the idea that you could take carbon dioxide and just join the 3 carbon dioxides together, or 6 in one go, and make glucose, how could you do that? You can't join two carbon dioxides together. You have to join it to something else. And the other thing is that in the process of the incredible enzyme that does all this, rubisco, is thought to be the most abundant protein in the world, because the leaf comprises about 25% of the weight of leaves, and it's involved in photosynthesis, of course. So all the plants in the world have rubisco. It's probably the most abundant protein. Enzymes are proteins on the planet. But the thing is that it uses energy that's produced from the light cycle. So photosynthesis involves chlorophyll, and the chlorophyll harvests the light and produces various energy-rich compounds. And these energy-rich compounds are then used to produce, to incorporate carbon dioxide into sugars. But the thing is that that's a process that involves several steps, 4 steps, and 3 of them involve these energy compounds. If you did that, and that's one for one carbon, so you go around one carbon, add another carbon, add another carbon. If you added 6 carbons together in one, if you could do it, and all that energy would be required to do it, you'd have a heat problem. That's just ridiculous. These people come up with these ideas that something's bad design. But another angle which they claim is bad design is a C3 photosynthesis, which is the one that most people know more about, because oxygen competes with the active site on the rubisco for carbon dioxide, and because of that, it actually causes the thing to go what they call photorespiration, so release of carbon dioxide rather than incorporating it, and also it regenerates the energy compounds that receive the energy from the light cycle. But this is a loss of carbon dioxide, or a loss of carbon from the cycle, this photorespiration, and it's accelerated when you have high temperatures, very high light and very high temperatures, and that sort of thing. So they claim this is inefficiency, and on the surface of it looks like it is. But the thing is that it's actually a protective mechanism. So when there's light's very strong, the temperatures are high, and there's an excess of energy being produced, this is a way of actually soaking up the energy so you don't damage the photosynthetic system. And the other thing is that if you look at the carbon dioxide levels in the atmosphere today, plants are actually starved of carbon dioxide, contrary to the hype about global warming. We have very low levels of carbon dioxide in the atmosphere today compared to what's been in the past. Pre-flood, we're looking up to 15 times the amount of carbon dioxide we have today. So plants are actually designed for much more carbon dioxide. And if you have 15 times the amount of carbon dioxide, then the photorespiration is virtually nil, because you've got plenty of carbon dioxide to soak up the energy and produce sugars and soak up the energy from the light cycle. So we'll talk about C4 plants later, but C4 plants have a way of concentrating the carbon dioxide and delivering it to the rubisco, so that there's no lack of carbon dioxide at any stage. So it saturates the rubisco a bit like 15 times before the flood. The plants actually concentrate it so that they deliver it to the rubisco with a concentrated form, and then the oxygen doesn't compete for the site on the rubisco, and you don't have the problem of photorespiration. So the C4 plants don't have a problem with the photorespiration, but they're also better adapted to high temperatures, C4 plants, because they don't have this photorespiration issue. So C3 plants tend to be better in cooler conditions, temperate conditions. You think of wheat as a C3 plant, whereas maize or corn is a C4 plant, and it's tropical, and whereas wheat is temperate. So this difference in the C3 and C4 plants is due to the adaptation to different temperatures, and also the fact that the plants clearly were designed to operate under much higher carbon dioxide levels, in which case you don't have a problem with the photorespiration. So the people make these charges that things are inefficient or whatever, or they could do better. They don't know what they're talking about.
That's really amazing. If you don't mind, I actually would like to ask a follow-up question about the carbon. So I'm wondering, like, would it actually be better if there was more carbon dioxide for the plants?
That's correct. In fact, there was a paper in Nature, which is the world's top science journal, about 2 or 3 years ago now, where they looked at the effect of the increased CO2. So the CO2 was increased from about 280 parts per million to about 410 at present over the last 50, 60 years. And at 280 parts per million, plants were really starved. But at the 410, this article in Nature, they looked at the effect of the increased CO2 on the plant productivity worldwide. And they came to the conclusion that 30% of the biomass production in the world, in other words, that's food and fiber from plants, 30% was actually due to the extra CO2. So the extra CO2 is actually greening the earth. In fact, when you have more CO2, you need less water, particularly for C4 plants and desert plants like cactus and things like that. Cactus are also C4 plants. And so they need less water when you have more carbon dioxide. And so you get a greening of the deserts. And this is happening as well. So if you want to be a greenie, you should be barracking for more CO2.
Wow, I didn't know any. That's pretty amazing. And then if we can put a link to those papers maybe in the description of this video. I'd appreciate it if you could send those to me when we're done with the interview. Thank you so much. And thank you for re-answering that question from so long ago. That actually reminds me of another lesson that I was taught in biology. So we had to study what was called the endosymbiotic theory. So Dr. Batten, I remember after I was taught that in class, I went home and I looked it up. I looked up the word endosymbiosis on creation.com, and I found your article that said, did cells acquire organelles, such as mitochondria, by gobbling up other cells? You wrote, the endosymbiont idea was severely dealt with in the 70s and early 80s, and should have died. But what else is there for the evolutionist? It's very much akin to chemical evolution. Anyone who knows a little bit of the biochemistry involved in the most basic of bacteria knows that the formation of a living cell from chance chemical reactions, even in highly controlled/contrived Miller-type experiments, is absolutely impossible. So in this article, you also pointed out how you would expect to find similarities in many of the genes for photosynthesis or respiration. Dr. Batten, can you expound more on why the endosymbiotic theory is false and why you expect to see similarities between respiration and photosynthesis?
Well, to go back to why the endosymbiosis doesn't work as an explanation of the origin of eukaryotic cells. So this is the idea that prokaryotes are things like bacteria, and they have a relatively simple structure. They have a membrane enclosing basically cytoplasm, and they have a single loop of DNA, and they don't have organelles like chloroplasts and mitochondria and things like that. So the organelles are membrane bound. So superficially they look like bacteria, a bacteria inside another cell. So the idea was that, well, eukaryotes arose from prokaryotes by one prokaryote engulfing another. And so the other one that was engulfed became the organelle, like a mitochondrion, or if it was a photosynthetic bacterium, it would become a chloroplast. And so this is the just so story for how eukaryotic cells, which have a membrane around a nucleus and chromosomes and organelles, arose. But that idea was possible back then, like 30, 40 years ago, but the knowledge of the structure of both prokaryotes and eukaryotes is such today that it really doesn't stack up. Just a few of the problems. One problem is that there's apparently no example of a prokaryote engulfing another prokaryote. So the idea of phagocytosis, that you can gobble up another one, it just doesn't happen. So eukaryotes do that. They can gobble up a bacterium, but bacteria don't gobble up bacteria. So that's the first problem. But also, prokaryotes have their ATP synthase motors. So ATP synthase is necessary for all life. Even anaerobic bacteria have it to go in reverse. But it's in the outer membrane. So the ATP synthase has to be bound in a membrane to separate the protons. So you have to have a proton gradient to drive it. And to separate the protons, you have to have a membrane. So that in bacteria, it's the outer membrane. In eukaryotes, it's the membrane of the mitochondrion inside the cell. So if eukaryotes arose from one prokaryote engulfing another, there should be ATP synthase in the cell membrane, but there are none, no sign of them. It's also, mitochondria are the power plants of eukaryotic cells, and 33 of the 81 proteins identified so far in humans in the ribosome. So the mitochondria have their own ribosomes. They're called mitoribosomes. The ribosomes are where you manufacture your proteins. So ribosomes take the messenger RNA translated into proteins. So they line up the amino acids in the correct order as specified by the messenger RNA. So there's ribosomes, there's special ribosomes for the mitochondria. And so 33 of the 81 proteins involved in the ribosomes of the mitochondria in humans have no analog in bacteria. So they're completely unique. So if the eukaryotic cell arose from prokaryotic cells by this process of endosymbiosis, how come we've got all these unique proteins? They were uniquely created. In fact, just one of those proteins is enough to sink the whole idea. In fact, one of those proteins is enough to sink the whole idea of evolution. So, because not even one protein could arise by evolutionary processes. So the other thing is the mitochondrial genome. So the mitochondria has its own loop of DNA, which specifies how to make some of the components necessary for the mitochondria, but not all the components. In fact, over 90 proteins are required to produce the mitochondrial ribosomes, but nearly all of them are supplied by the nuclear DNA. So they're not actually specified by the loop of DNA in the mitochondria. So there's incredible linking of the information in the mitochondrial DNA with the information in the nuclear DNA. And of course, the other thing is that chromosomes, for example, in eukaryotes have a structure with histones, and they have an actual chromosome structure, they're not just a loop of DNA. And no bacteria have anything like that. So there's so many profound differences. Actually, there's a new article by Dr. Jerry Bergman in the current issue of the Journal of Creation on the endosymbiosis idea, brings it up to date. Worth reading. Those who subscribe, it's actually fairly good value. It's full colour, comes out 3 times a year. The Journal of Creation, the current issue, has a major article about the endosymbiosis idea and why it's falling on hard times.
That is really amazing. And so another part of that article I remember is you said that when you're looking at photosynthesis and respiration, you were expecting to see some kind of similarity. Is there like a reason for that? Is it like because of common design for that similarity?
I think the similarity is really mainly just in the fact that they are membrane-bound organelles, because you have to isolate what's going on from the rest of the cytoplasm of the cell, otherwise it just wouldn't work. So to that extent, there are similarities, and they're membrane-bound, and the membrane of the mitochondria, you've got the ATP synthase, many of them in each mitochondria, and all the other things that go with it. There's proton pumps and things. It's just amazing. And a similar thing, the chloroplast has to be isolated from the rest of the cell for it to function the way it does. So to that extent, there are similarities, but there's not a lot of similarities in the enzymes and things that are involved. Yeah, so.
Thank you so much for explaining that. I think that all of the viewers or the listeners should definitely subscribe to the Journal of Creation. I have the first copy I got, I remember, was when I went to go see Dr. Carter, and it has an armadillo on the cover. It was really, really amazing.
I remember that one, yes.
Yeah, like it's very beautiful, and we'll put a link of it in the description. That way people can subscribe to it.
It's the in-depth journal. I mean, it's where all the debates occur. If you want to go into the, and like you, Christopher, you're interested in all the ins and outs of things. And if you just want to skim over the surface, then the journal's not for you. But if you want to go into depth, the journal's certainly for you.
Thank you so much, Dr. Batten. Well, since in our last question we touched a little bit on photosynthesis, I think it would be interesting to cover a plant that has been said to be vital for our own existence. According to Education, Environment, Adventure, we as humans cannot survive without trees and other plants because we breathe in oxygen and breathe out carbon dioxide. Dr. Batten, in your article, Tree Ring Dating, and that's Dendrochronology, you wrote, recent research on seasonal effects on tree rings and other trees in the same genus, the plantation pine, Pinus, I think it's Ratia, has revealed that up to 5 rings per year can be produced, and extra rings are often indistinguishable, even under the microscope, from annual rings. As a tree physiologist, I would say that evidence of false rings in any woody tree species would cast doubt on claims that any particular species has never in the past produced false rings. Evidence from within the same genus surely counts much more strongly against such a notion. Creationists have shown that the biblical kind is usually larger than the species, and in many cases, even larger than the genus. See my article, Ligers and Wolfins. What's next? Dr. Batten, this article was written back in 2006, so in your assessment, has the case for the evolutionary timeline from dendrochronology grown stronger since then, or are there more problems with this dating method that have arisen?
Well, they've certainly extended the the timeline of the calibration and carbon-14 dating. So that's what they've done. They've tried to do is, and what they've endeavoured to do is extend the carbon-14 calibration. So carbon-14 dating, when you can calibrate it with historical dates, it can be useful. But once you get past historical dates, where do you go with it? So they've tried to use tree rings to extend it. And the initial attempt of this was with the bristlecone pine in the United States. And the oldest bristlecone pine is about 4,700 years by counting the tree rings. Of course, that would put it pre-flood, strict biblical chronology. But of course, it would only have to be shortened by a couple of hundred years, which is not much in the 4,700, by some extra tree rings. In other words, not assuming 1 ring per year. And of course, in the post-flood world, which was quite warm and humid initially, and then the development of the Ice Age about 500 years, peaking about 500 years after the flood, and then waning very quickly over about 200 years. But the initial phase, you've got very humid, lots of rain, and you've got conditions where multiple tree rings would almost certainly have happened in the early days. So they might not be happening today, but we look at the biblical history and we think there's good reason to believe that would have been happening back then, and particularly when the trees are young. And that work in radiata pine, for example, when the trees are young and they're closely planted, like in a forest designed for wood production, initially when they're not competing with one another, you get multiple rings. But as the trees grow and they start to compete with one another, then they start to not have multiple rings. So young trees, after the flood, we would expect to have multiple rings. That would reduce the age. So that's the oldest living tree is about 4,700. And how do they get earlier than that? Well, they've found pieces of wood lying on the ground, and they think, oh, these might be older than the living tree. And so they carbon date them. And they assume a straight line backwards with the carbon dating, and then place that piece of wood according to that assumed straight line backwards extrapolation, and they place it there, and then they look for cross-matching of tree rings with the living tree. So you get one which is supposedly, say, 4,800 years old, 4,900 years old, and then you cross-match the tree rings with the living tree and try and find a spot where they match up. And then you then extend it into the past. That seems reasonable on the surface of it. But the problem is it assumes that the carbon dating is correct, and you're trying to calibrate the carbon dating. So it's a totally circular process. And as one of the researchers, Yamaguchi, the reference is in my article on this, he actually showed that there are multiple places where you can get cross-matching of a tree ring. So there's no unique spot where you could say this definitely links up here. It's only because of the carbon dating that constrains it that you can actually say this is where it links up. In fact, he showed that there are other places where you get an actually better match in the tree ring structures, tree ring pattern, than where the carbon-14 says you should put it. So the other thing is that John Woodenruppy, he wrote a paper, published a paper in the Journal of Creation on this, because he went and did some hands-on stuff with the bristlecone pines, and he remarked in the paper that the pieces of wood which are supposedly older than the oldest living tree are supposed to be different ages, but he said they all look much the same. There doesn't seem to be any indication of something being old or young. In other words, what does it all mean? So in Europe, they've got three tree ring chronologies they've developed, and they try to do those independently of the bristlecone one, because that would then corroborate the bristlecone one. But what they found was they actually disagreed. So they then basically fixed the European ones to the bristlecone pine one. So they're not independent at all. So it's all a bit of magic and hocus pocus, really. So I'm very skeptical of the whole process, and I have no problem with. If you look at not just bristlecone pines, and by the way, if you compare one bristlecone pine tree with another, the tree ring structures are actually quite different, even though they grow near one another. So this matching thing is questionable. But some of the oldest trees, in fact, there's an article written by Gary Bates, called Methuselah trees or something like that on creation.com. It was in Creation magazine. And he looked at the oldest living trees, and there are no old living trees that predate the flood or the bristlecone pine. So isn't it funny that you look at the oldest trees and they're like thousands of years old? They're not tens of thousands of years old. Is there any reason why? Yeah, well, Noah's flood would explain why there are no trees that are 10,000 years old, no living trees. So, yeah, a lot of people get hung up on this dendrochronology. They think it's rocket science, but it's quite circular.
Dr. Batten, thank you so much for covering the dendrochronology dating method. There's actually another plant-based problem to the evolutionary dating method. Dr. Batten, in an article called Pollen Problem, you said that researchers have reported finding several types of pollen from flowering plants in dinosaur rock, the Middle Triassic period. On the evolutionary timeline, this puts the origin of flowering plants 100 million years earlier than previously accepted. Consequently, the dioramas in museums and children's books on dinosaurs ought to show flowering plants living at the same time as dinosaurs. To date, however, you'd be hard-pressed to find any such plants illustrated, reinforcing the false idea that dinosaurs lived in an entirely different other world. Dr. Batten, this is a two-pronged question. Has this pollen research stood the test of time, and if so, how can we use details like this to challenge the common narrative of millions of years?
Now, this is just one example. In fact, they've found coprolites, fossilized dung, and coprolites of dinosaurs. They've found trichomes of grasses in them. So these are grasses are flowering plants. And this was a big surprise to the evolutionary researchers because, again, they've got this idea that dinosaurs live in an entirely different world before flowering plants. And so you see them with cycads and palms and that sort of thing, but you don't see them in the dioramas at museums or even Jurassic Park, Jurassic World and things like that. You don't see them with modern flowering plants, but in fact, those modern flowering plants were there. But the pollen in the Triassic is just one example of many. There's far worse problems for the evolutionary timeline in terms of pollen. For example, the Roraima formation mountain in Venezuela. Now, this is old work, but it really was published in Nature, and there are multiple re-investigations of this stuff to try and figure out what was going on, because they found plant pollen in this rock that's supposed to be at least 1.7 billion years old. In other words, Precambrian. Now, this means that the pollen is 1.4 billion years older than when plants are supposed to have appeared. So there's two approaches to this. One is, the rocks were misdated. They can't be Precambrian. They must be more recent. So they went and looked at the geology, and they couldn't get away from the geology. They had to stick with their 1.7 billion years or Precambrian dating of the rock because of the sequence and everything. And in their scheme of things, you can't just redate things. They're in a sequence of things, and if you redate those, then it throws everything else out. So the system means they've got to stick with that date. So then, maybe the pollen was misidentified. So check that. Maybe it was maybe there were cracks in the rock and they washed down there from recent plants. No, that didn't work. So in the end, they just basically shrugged their shoulders and said, "We don't understand." But the problem is the age. The problem is the dating. The problem all along is the age of things. If they could look at it and say, "Well, these rocks aren't that old," but they can't do that. So, and the other one was Grand Canyon. This was done by creationist researchers, published in the Society Journal, Creation Research Society's journal, the quarterly. And the initial research showed this pollen in Precambrian rocks in the Grand Canyon. And other creationists said, that can't be right. So even creationists doubted it. So they went and did really careful work. They went in winter when there's no pollen floating around the air. So there's snow on the ground, there's no flowering, there's no plants flowering, there's no pollen in the air. They put out slides with sticky surface on them while they were working to make sure that there was no pollen blowing in from Texas or something, or from Mexico. So they left those pollen slides there, and then they looked at those later to make sure they hadn't collected any pollen on them. And they also made sure that there were no, when they sampled the rock, they made sure there's no cracks in the rock. So there's no way that there could have been washed in through the cracks or something like that. There was such care taken to make sure that there couldn't have been contamination. And then the people who were skeptical of the pollen being found said, "No, there's pollen there." And this is not a problem for creationists, of course, but But because it was would be so controversial in the wider scientific community, these absolutely incredible steps were taken to make sure there's no contamination, properly identified, etc., etc. So there's quite a number of examples of pollen being found where it shouldn't be. And the problem is the dating of the rocks. That's what the problem is. And of course, if the rocks, say the rocks of the Grand Canyon, including those Precambrian rocks, if they formed during Noah's flood in the space of a year, you can see how the pollen could have been separated from the plants that it came from by the process of the sorting action of water during the flood. And so you find, for example, with the Morrison Formation in the United States, you find dinosaur remains, dinosaur fossils, but you don't find their food. So what do they eat? If this is a burial of an era of Earth history, a recording of an era of Earth history, where is the food? Because nearly all the dinosaurs had to be vegetarian. They weren't all T-Rexes. You have to have something, vegetation to feed the vegetarians, to feed the carnivores, So where's the vegetation? And of course, again, there's a problem with the disbelief in Noah's flood. Noah's flood would explain why you could separate the dinosaur fossils, the dinosaur remains from their food sources, because of course they would have different buoyancies and things, and the trees and things that they ate the leaves from would have been transported quite differently to their dinosaur remains and buried quite differently.
Dr. Batten, that is really awesome. And while we're on the subject of these plant-based problems for elements of the evolutionary theory, I want to highlight an interesting article you wrote on creation.com called Eat Your Brussels Sprouts. This article, it highlighted mutations in the Brussels sprouts and its implications for the evolutionary theory. The article says, for example, in the common heading cabbage, only one bud at the top of the stem, the terminal bud, produces leaves. There are other buds, one at the base of each leaf, but they do not develop unless the terminal bud suffers damage, say, by an insect eating it. Then one or more of these auxiliary buds develops. In Brussels sprouts, the plant fails to suppress the growth of the other buds, and so each one becomes a little cabbage. It also has an elongated stem compared to the cabbage. Such mutations would not increase the complexity of an organism, rather they mess up some part of its functionality. So Dr. Batten, are mutations, like the ones you mentioned in relation to the Brussels sprouts, evidence for or against the evolutionary theory?
Well, evolutionary textbooks and courses tend to downplay the role of mutations in their theory. But mutations are the only mechanism they have for generating the new genetic information required for evolution to progress from more simple organisms to more complex organisms. So they tend to focus on natural selection, because you can find lots of examples of natural selection, but you can't find examples of mutations that actually generate new genes, new enzymes, which are needed. So all the mutations mutations are accidental copying mistakes. So when the information from an organism is passed on to the next generation, some mistakes can be made in copying the information. Those mistakes are called mutations. They're like when you're typing on your computer and you hit the wrong key and you end up with a typo. So mutations are basically typos in the DNA. So you expect that typos would actually tend to mess up the information. They wouldn't create new information. You wouldn't shut your eyes and bash away at your keyboard hoping to generate a novel idea. Ideas come from brain, from intellect. They don't come from random processes. So you can mess up things. There are many ways you can mess up things. There are very few ways you can make them better or improve them. And there are mutations that are beneficial. That's not the issue. But they're beneficial by breaking things. And a classic example is some beetles that were blown onto a windy island, and when they took to the air, they got blown into the sea. But some beetles were born with a mutation such that their wings didn't develop properly, and so they couldn't fly. And so the beetles with the defective wings had an advantage over the ones that had normal wings. And very quickly, natural selection selected the ones that had the defective wings. And of course, the others died being blown into the sea. So you end up with a population of beetles on the windy island which have defective wings. So there's a beneficial mutation, but it's breaking something. It's beneficial to survival under those circumstances, but it's breaking something. So that's the problem for evolution, is the mechanism doesn't work. And 100 years ago, when mutations were discovered, they were heralded as the mechanism for the new information. And so that then heralded an era of mutation breeding. So you've probably heard that Drosophila, which is the ferment fly, and all biology classes talk about Drosophila. Well, Drosophila were irradiated with gamma radiation to cause mutations. And this process was expected to generate superflies with new features. All those mutations did was break the existing features. So you end up with flies with no wings, or flies with defective wings, or flies with different defective eyes or defective colours, or no one ever found anything which indicated it was on the way to becoming some superfly. So, and virtually every possible mutation's been created in those Drosophila flies, and nothing to give encouragement to the idea that mutations are the mechanism for generating all the new information. So there are all sorts of mutations. You can add DNA letters. You can take them away. You can lose them. You can duplicate them. But none of the processes generate the code required for a new enzyme. And that's the problem, because until the enzyme functions, there's no way natural selection could select it anyway. So there's a huge problem with this whole story. So consequently, mutations are played down as being the mechanism for evolution, but they are the mechanism. Without mutations, there's no evolution. Natural selection is not evolution. Natural selection operates in today's world. It preserves the fit as against the unfit, but it doesn't create new features. All natural selection can do is select from existing features. Like, for example, you've got beetles with wings, beetles without wings. Natural selection can select, if you like, the ones without the wings under the windy conditions, but it can't create the ones without wings. It can only select from what's there. So it's a big con of the evolutionary proselytizers to claim that natural selection equals evolution, but it hoodwinks a lot of young people into thinking that evolution is a valid scientific theory that explains how microbes changed into microbiologists, and everything else on Earth came from microbes by the process of evolution. So mutations are really a problem for evolution. The other problem with the mutations is because they're wrecking things, and there's so many of them, they're actually causing the deterioration of the genetic code in organisms, and this is happening in humans. And Dr. John Sanford has written about this in a book on genetic entropy. You've probably spoken to him. And the secular geneticists recognise that the number of mutations in humans is such, in each generation acquires an extra 100 mutations. Natural selection can't get rid of them, and so they're accumulating. And so what natural selection could get rid of, if we had one, say, if we average one new mutation per generation, natural selection could slow down the deterioration. But when you get 100, you can't get rid of. No offspring will be born with fewer or the same number of mutations as their parents. Every child will have something like 100. It might be 90, it might be 80, but many, many more than the parents. So it's inevitable that deterioration occurs. And so we find the things that are degenerating, they're not on an uphill trajectory like evolution would claim.
Dr. Batten, thank you so much for covering how mutations are not evidence for the evolutionary theory. This actually reminds me of your comments in the article, "Plant breeding is not evolution." plant claims hard to digest. In this article, you wrote, I have no problem with the concept that mankind has selected and developed various crop foods to make them nicer to eat or easier to cultivate, but sometimes not as nutritious or healthy. This has nothing to do with evolution, as there is no evidence that the processes involved the natural addition of new genetic information to the species involved. The natural processes of mutation and natural or artificial selection do not add new biochemical pathways to living things. Dr. Batten, what is a biochemical pathway? And can the evolutionary theory account for the emergence of them?
The biochemical pathway is where you get a sequence of transformations of a chemical. For example, if you want to generate hemoglobin, for example, you have to start with simpler chemicals and join them together in a sequence of events, which then arrives at the functional hemoglobin. And that sequence of events can involve, a dozen enzymes. And there's no point in having just one of the enzymes. If you haven't got all of them working together, you don't get the product. And you can buy a chart from the chemical companies, the biochemical companies, the companies that sell biochemicals have these charts, and you look them up on the internet, like Sigma Pharmaceuticals, for example, or one of these, and the chart shows a simplified diagram of the pathways in a human. So it shows you all the different enzyme steps involved in generating estrogen, for example. Now, plants are incredibly complex because they just take carbon dioxide, water, and some minerals from the soil, and they generate all the biochemicals of life. Now, we actually eat the plants and get a lot of the biochemicals already made from the plants. So all the vitamins and amino acids, we don't make amino acids, we get them from what we eat. So we get them from either animals that eat plants, but ultimately they come from plants. So all these things are involved in biochemical pathways with multiple enzymes involved. And unless, with most of the biochemical pathways, there's no actual usefulness of any of the intermediate products. Okay, so there's no way that, even if you could get one of the enzymes to transform A into B, there'd be no benefit from having B, because it doesn't do anything unless it's transformed into C and transformed into D and E and F and goes through to the end product. So you need all the steps in the pathway for it to be any use. And there's no way that evolution can even get one enzyme, let alone all of the enzymes working together. So the thing's dead in the water.
So, Dr. Batten, that was a really interesting explanation, and thank you so much for explaining what the biochemical pathways are and how they relate to evolutionary theory. I think that we should also now take a look at the capacity human ingenuity can play in the origins debate. So I've heard a lot of talk, especially recently, of claims that humans are basically like a step away from creating life in the lab. So some believe that the origin of life problem was solved at the Miller-Urey experiment. There was one of your articles, it was called, What? No Potatoes? Where you explained how the lack of genetic information in the Irish potato crop caused them all to succumb to the potato blight. You said, if random copying mistakes, mutations, originally generated all the information, surely it should not be too hard for highly intelligent scientists to create the required genes for breeding new, improved varieties. However, with all that we now know about genes, no one can yet create a gene, for example, for rust resistance, from scratch. Plant breeders recognize that the information in the genes of plants is irreplaceable. Dr. Batten, what do you make of the fact that not just the creation of life in the lab, but even the creation of a gene from scratch is beyond our reach?
Yeah, in fact, there are a couple of seed banks in the world where seeds of many, many thousands of species of plants have been stored to preserve their genetic information, so that if they do go extinct in the wild, then the seeds will be there to be able to regrow them, because plant scientists recognise that we cannot create those genes and things. We can't even begin to actually imagine what we need to do to create them. So the Venta, for example, the lab just recently announced that they'd synthesized a bacterial cell, and it was made headlines around the world, almost as if they'd created life in a test tube, which they hadn't done. But they basically copied an existing bacterium. And about 10 years ago, they did the round one of this, and they proclaimed that they'd synthesized life, but they'd copied existing life. In fact, they'd taken an existing cell and taken DNA out of another that they'd created, copying the DNA of a real thing. They copied the DNA and they put it into a cell and claimed they'd created life, basically. Well, the media claimed that. But in fact, they're copying life. But their interest in doing that is a very practical interest, in that they want to get a bacterial cell or a microbe that they can modify to produce biochemicals. So, for example, you could get it to produce human hormones or drugs that might be useful for curing disease and things like that. So it's a worthwhile exercise. So, and what the question was, how simple could they make something which would be viable? So they took the simplest known living thing, which is actually a parasite, can't live by itself, and they started removing genes from it to see how simple they could make it and still live. Well, 10 years ago they did that. But then just this recent announcement was that, although actually what they did 10 years ago wasn't really viable, because when the cells divided, they didn't divide properly. So they now found they needed an extra 10 genes had to be added back into this microbe for it to actually produce viable cells. So they're actually adding genes back in to make it viable. And where did they get the genes from? Did they manufacture those genes from their imagination? No, they took them from the original organism. It's incredible that intelligent scientists are scratching around trying to understand how an enzyme works, and applying all their intelligence using supercomputers and things to try and predict folding and all sorts of things in enzymes. Because the enzymes are three-dimensional structures, and they're produced as a one-dimensional structure, in a sense of a string, and that string has to fold into a three-dimensional structure, and the folding process is incredibly complex. And even just the simplest enzymes stretch the ability of a supercomputer to predict the folding. And yet these same scientists blithely believe in evolution, that random accidental changes generated these enzymes. This just doesn't add up. Just doesn't add up. In fact, I was talking to a molecular biology student, PhD student at the University of Melbourne, after I gave a talk down in Melbourne. And he said similar. He said, I can't believe my colleagues who study this incredible stuff every day and yet ascribe it to evolution. He said, it just doesn't add up. It just speaks of incredible intelligence, the intelligence of our creator. You know, we should be saying, wow, how incredible is our God? How incredible is our creator? But now they say, wow, isn't evolution amazing? Yeah, so things like rust resistance, all sorts of things. We just don't know enough yet to be able to even begin to imagine how to create a gene which would produce something useful like that. So we're so dependent on, that's what plant breeding, even genetic modification is still taking a gene from something else and putting it in different species. So for example, with the process of generating cotton, which has the Bacillus thuringiensis toxin in it. So Bacillus thuringiensis is a bacterium that will kill the bollworm Heliothis. And and you can spray the toxin on the crop to kill the bollworm, but then you could actually genetically modify the cotton to produce the toxin, so that if a bollworm hatched out on the surface of the cotton and started eating the cotton, it would get the toxin and die. And this has resulted in a huge reduction in the amount of pesticide being sprayed on cotton. Very beneficial genetic modification. But it's not scientists inventing what they put into the cotton. It was taken from Bacillus thuringiensis. It already existed. So the genetic modification is taking something from somewhere else that we've discovered and putting it in a different species. That's all it is. It's not, they're talking about gene editing, and the gene editing, they're even talking about doing this on humans. This is questionable morally, but there's been some attempts to do this for some forms of blindness and things, and with some success. But the gene editing is actually fixing just a mutation which has caused the blindness. So, for example, a particular gene has a particular letter, wrong letter, and the gene editing is to edit that and put the right letter in there. So it's fixing up something that mutations have caused rather than actually inventing something to start with.
So, Dr. Batten, while it's true that evolutionary scientists, they've not been able to create a gene from scratch, evolutionists will sometimes point to creationists with criticisms of our own conception of the original created kinds. You touched on this subject in your article, C4 Photosynthesis Evolution or Design. Dr. Batten, the article says, some species that exhibit both C3 and C4 forms are able even to switch from one to the other during development. This suggests that maybe the C4 chemistry is latent in C3 plants or is suppressed by some means. These developments underline just how cleverly the original plants were created, with built-in latent capacity for adaptation to a wide range of environments. It'd be interesting to see the details fleshed out. Dr. Batten, this is another two-pronged question. What's the difference between a C3 plant and a C4 plant? And may you please give the audience an idea of what the original plant kinds were?
Oh, big question. So we touched upon C3, C4 earlier. So C3 plants are things like wheat and your temperate plants, which just take in carbon dioxide into the leaf through the pores called the stomata, and the air circulates around the photosynthetic cells and enters the cell, and it's directly taken up by rubisco, that enzyme we mentioned earlier that's involved in the so-called dark reaction, and that's incorporated into sugars. A C4 plant is quite different in the leaf structure, in that it takes in the carbon dioxide through the stomata just the same, but then it incorporates it into say malic acid, for example, a malate, which is a C4. And so it incorporates the carbon dioxide into this malate, and then the malate is transported to the photosynthetic cells. And the photosynthetic cells don't have air all around them, they actually are enclosed. And then there are three enzymes involved that actually release the carbon dioxide from the malate, and it presents it to the rubisco in a concentrated form. So, in other words, the rubisco is then running at maximum capacity rather than being starved of carbon dioxide. And consequently, these tropical plants like maize and things like that have this C4 chemistry in their leaves. Now, it used to be thought that there was this big chasm between C3 and C4 plants, but then it was discovered that the number of C3 plants that were looked at actually have the C4 chemistry in their roots. So this actually stands to reason, because there's quite a lot of carbon dioxide in the soil because of the aerobic activity there, microbes and worms and all sorts of things generating carbon dioxide in the soil. So lots of carbon dioxide in the soil. And here it is, the C3 plants have the carbon dioxide absorption system in the roots to make malate, and then it's transported up to the leaves where it's released to feed the photosynthetic activity in the leaves. So the plants are actually taking carbon dioxide out of the soil and then transporting it in malate up to the leaves for photosynthesis. So then it became clear that C3 plants actually have all the chemistry for C4, but it's in the roots and in the stems rather than in the leaves. And so the difference between the two now is much less than it was thought to be. The evolutionists even proposed that because there's no pattern to the existence of C4, it's all over the place. All different families of plants have it, and different, even some families have C3 and C4 plants. And we would say the families derived from the original created kind. So there's different C3, C4 in the same family, even the same genus. And in fact, as I mentioned, some plants can change from one to the other depending during the development and depending on the environment in which they develop. So it appears that all plants originally had the C3 and C4 chemistry. It's just that some have adapted to more temperate conditions, and some have adapted to more tropical conditions through accentuating the C4 in the leaves or C3 in the leaves, depending on the environment, depending on the growing conditions of the plant as it grew up, etc. So it seems that C3, C4 is not something that's distinctly different, but it's basically in all the plants. And so it's really got nothing to do with the identification of original kinds. So, for example, when I worked on lychees, there's a whole lot of different plants in the Sapondaceae, which is a lychee family, and that includes things like the rambutan, the longan, the palosan, and there's a number of different ones. There's a native plant in Australia, which is in the same family, and they all have clearly similar characteristics. They're pinnate leaves and the fruit with flesh and single seed in the middle and different things. So each of the families of plants has its own particular characteristics, and it seems, generally speaking, that with plants, that the family is the level of the created kind. And so what we see today with the species and genus, different genera and species within a family are derived from an original created kind. But there's a number of papers on, they're called baraminology. Bara is create, and baraminology, baramin is created kind, and baraminology is the study of the created kinds. And there's one in the journal Creations on the website on the origin of grasses, for example, and all the different species of grass. And we can look at hybridizing and things like that, which show that they were originally one created kind. I was involved in creating a hybrid of the longan and lychee with different genus, members of the same family, which illustrates that they came from an original created kind, which was probably in between the two that we're talking about.
Dr. Batten, as we bring this interview into its closing segment, I want us to take a look at the bigger picture of the origins debate. Dr. Batten, Creation Ministries International started a Question Evolution campaign. The campaign centered around 15 crucial questions that the evolutionary theory does not have the answer to. Obviously, this campaign was met with criticism from evolutionists, and in an article called How Evolution Has Harmed Science and Society, you responded to one such evolutionist by saying, "Many of these questions are fundamental to evolutionary theory, and yet there are no reasonable answers. That is, evolution is an exercise in bluff, of pretending to have answers where there are none to the big questions. There is also a pretending that science needs evolution, when in fact it impedes the progress of science, as money is wasted on chasing phantoms like the naturalistic origin of life. Research has only increased the case for it being impossible." Dr. Batten, what do you see as the most detrimental harm the evolutionary theory has caused to science?
I'm actually in the process of developing an article where I'm listing and annotating, commenting on, I'm up to 35 so far, 35 ways in which evolutionary thinking has harmed science. But probably as a heuristic, as a way of thinking, the idea that living things are just cobbled together by random processes, sort of by natural selection, causes people to think that things are simple. And with this, a bit of application of intelligence to the situation, we should be able to improve the situation. But it's completely naive. And I think it's a dangerous view that thinks that just by a little bit of tinkering, we can improve things. If they took the view that things were created by an incredibly intelligent designer, then there would be a different approach, which would show respect for what they're researching, and also expect to find sophistication. And but the contrary is they expect to find cobbled together things, not sophistication. And consequently, it's an impediment to research across the board. Probably one of the most detrimental views was the idea of junk DNA. So this idea was that evolutionary thinking needs lots of junk DNA. There's three reasons why evolution needs lots of junk DNA. One is that mutations can't possibly generate 100% functionality. That would be unbelievable. So there must be lots of non-functional DNA because that's what mutations would do. Secondly, there has to be lots of non-functional DNA for there to be experimental room for mutations to generate new information. And thirdly, because there are so many mutations, if all the DNA was functional, those mutations would be a problem for the viability of the organism. So therefore, they decided that 98% of the DNA was junk, because that would mean 98% of the 100 mutations were of no consequence. So only 2 mutations then, out of the 100 that were known, only 2 would be actually important or cause any problem. And therefore, you can then start to imagine that humans have been around for millions of years, coming from apes and so on, and that natural selection could conceivably slow down this deterioration to a point where you could believe in evolution. So there are 3 reasons why evolution needed junk DNA and why it was part of the whole paradigm, and why the idea that it was not junk has been resisted even till today. So in spite of the evidence from the ENCODE project, which involved something like 135 labs around the world, or was it scientists? I can't remember. But there are many scientists involved around the world in different labs researching the function of the so-called junk DNA. And they found that, and you've got to look in all the different cell types. There's hundreds of different cell types in the human body. So you can't just look at one cell type, because what might not function in that cell type might be functional in another. So you have to look at all the different cell types. It's a big job. And anyway, the phase 2 of the ENCODE project came up with a figure that at least 80% of the DNA was functional, not 2%, at least 80%. And Ewan Burney, who was the spokesperson for the project, said that he believed that with further research, the figure would rise to near 100% functionality. This is a huge problem for the evolutionary story, a huge problem, because how can mutations create 100% functionality? And how can evolution progress if there's no junk DNA to experiment with? And what about the 100 mutations? Now, there are 100 mutations that are actually in functional DNA. So genetic entropy is inevitable. So the junk DNA, and it goes back further to the idea of vestigial organs. We had junk organs in our bodies left over from evolution. That was another one which resulted in so many dead ends and impeded progress in research. But yeah, over 100 organs were deemed as not important, useless, including the thymus and all sorts of things. So yeah, it's been a very detrimental heuristic for research.
So, Dr. Batten, thank you so much for outlining various different impediments that the evolutionary theory has actually brought on to science. And I'm looking forward to reading your article of the 35 different ways that the evolutionary theory has actually harmed science. You know, what's really interesting, though, is that when you contrast this with creation, creation actually gave rise to science. There's an article that you wrote called Creationist Contributions to Science, where you said, a fresh appreciation of the Bible's literal history in Genesis, including the fall of Adam, played a role in the development of the scientific method, the foundation of modern science. You went on to quote Peter Harrison, a professor of science and religion from Oxford University, who said, had it not been for the rise of a literal interpretation of the Bible and the subsequent appropriation of biblical narratives by early modern scientists, modern science may not have arisen at all. In sum, the Bible and its literal interpretation have played a vital role in the development of Western science. Dr. Batten, could you please expound upon those thoughts for us?
It's actually difficult to wind it back a bit. Operational science, which is where all the progress comes for understanding the world we live in and giving us what benefits for modern science, modern technology. Operational science, as in your introduction you said, comes from a Christian worldview. And so, in a sense, all scientific progress today actually rides on the back of a Christian worldview. Even the scientists who aren't Christians have absorbed a Christian worldview in their thinking and not even know it. They don't even know it. You can point to all the progress that's been made by anybody is actually because of the Christian worldview. The idea that we should do experiments. Why should we do experiments? It's back in pre-Reformation. The idea of the Greeks was that you could have pure thought. So you can have a thought experiment and you can work something out in your head, and then you can share that with somebody else. Now, that's a good idea, but nobody thought you should go and do an experiment and test whether that's actually right or not. So that's what Peter Harrison was talking about, the idea of the fall from Genesis, that we are fallen creatures, therefore our thinking is corrupted to some extent in terms of our ability to think perfectly logically. Therefore, we need to test our thoughts about the world. We need to do experiments. If you can't do an experiment to test it, then it's not science. And so this idea of testing things came about because of the Christian worldview, and modern science rides on the back of that. The idea that the world is real and that we around us, and it's not just an illusion. You know, if you have an Eastern mystical view, the world is an illusion produced by the spirits, by your ancestral spirits. So it's not something you can investigate. If a tree dies, there's no logical reason for it. It's the spirit left it. So because God is outside of his creation, and the creation is separate to God, it's not an emanation of the Spirit of God, but it's a separate creation. It's real, it's physical, can be investigated. And so there's a number of different, and we should investigate it because we have dominion over the creation in Genesis chapter 1. So we have dominion mandate to investigate and so on. And there's other ones as well. As in that article you mentioned, there's 6 different principles that undergird all of modern science. So all the modern scientific advances are actually due to the Christian worldview. All of it. So I don't have to point to specific examples of the way in which a creationist worldview has helped, but it would have helped enormously with the junk DNA. In fact, Dr. Carl Whelan, as a medical doctor back in 1994, is on record as saying, for many years, creationist scientists have said that the junk DNA is likely to be functional. So that's an example of where, if a creationist view of biology had been, rather than the evolutionary view, had been paramount in the thinking of people in the universities, then the junk DNA paradigm would not have been followed. They would have investigated and looked for the functions of the DNA. So, but in my article about creationist contributions, I mentioned 3 evolutionists who admit that evolution doesn't actually help much in doing practical science, in fact, at all. One was Mark Kirshner, Another was Larry Witham, and the third one was Philip Skell. And Philip Skell was a fellow of the American Academy of Sciences. So, and those three, you can see in that article what they say about how useless evolution is for doing real science. So the scientific progress that's happened is not due to evolutionary thinking at all. It's been held back by it.
So, Dr. Batten, we have now arrived at our final question. Your editorial, Jesus the Creator, is what I'd like to close with. You said the Bible clearly tells us that Jesus was present at creation, that creation happened by him. So he can hardly have been ignorant of the details, including the time frame. Dr. Batten, it's clear that Jesus is the Creator, and so how can Christians better embrace the creation message as a part of the Great Commission?
Anybody who tries to share their faith today will find quickly that many of those that try to share their faith with will respond by saying something like, oh, I don't believe in that stuff, I believe in science, or something like that. And what they mean is they've been indoctrinated in his evolutionary view, and they know full well that it doesn't actually correlate with the Bible. So therefore they've rejected it. I think they have a firm foundation for rejecting the Christian message. So we talk about creation evangelism, which is basically ploughing the ground to get rid of these stumbling blocks that people have. And they're very common. Barna research in the United States and in Australia have shown that it's the major issue for millennials, but it's also a major issue for all ages, that they think that science has done away with the Bible. And of course, it's to do with the history, which isn't science at all. It's to do with the age of the earth, what really happened in the past, and the Bible's account of what happened in the past. So we often then have to deal with this baggage that people have acquired to get rid of the stumbling blocks that they have for belief. And each person can be a bit different, and it can be simple for some and more complex for others. But usually there's something that people put their trust in. For example, they say, oh, there's all these ape men. Well, there's so much information you can share with them to show that they've been led down the garden path. And the response of some people, when you show them that, is anger, not at you, but at the system that's actually misled them. But they're then on the path to accepting what the Bible has to say about these things, and there's not too many steps from there to salvation. They're putting their trust in Christ to realize that there is a Creator who made us, to whom we will be accountable, and therefore I need a Saviour because I am not in a right relationship with my Creator. So the whole thing goes together. If God is not the Creator, there's no need for a Saviour, and Jesus is out of a job. So this is really where the rubber hits the road for many people. And so we as Christians need to really be informed about this, as says in 1 Peter 3 verse 15, to be ready with an answer for anybody who asks us for the reasons for the hope that is in us, but do this with gentleness and respect. And so the challenge to us Christians is to be prepared with the answers for people, and there are answers to so many things today that maybe there weren't answers even 40 or 50 years ago. And every day new scientific discoveries underline the truth of creation. It's actually, the evolutionary story is getting weaker and weaker, and yet the universities and the establishments have closed the door to considering there could be something wrong with it. So the way to deal with the contrary evidence is to shut it out. But we need to open that door on that evidence for the people we interact with and show them that you don't have to throw your brains away to believe the Bible. In fact, it's quite the opposite.
Dr. Batten, thank you very much for your time. It was an absolute joy. And to our listeners, thank you very much for taking the time to learn with us on current topics in science, where scientific discoveries are examined in light of the origins issue. Please share and subscribe to the Current Topics in Science podcast. It's available on iTunes, Stitcher, Google Podcasts, Spotify, and iHeartRadio. You can find Dr. Batten's books, DVDs, biography, and scientific research in the links in the description below. Thanks again for listening, and remember, the truth saves.